So now let's take a step down and get into the arterioles. So remember the structure of arterioles. They have that endothelial lining which prevents activation of blood clotting. They've got a really, really thick layer of smooth muscle and then some connective tissue. So their primary job, as indicated by their structure, is to step down the pressure. And the arterioles, remember, we go from a relatively small number of arteries to a relatively large number of arterioles. So the pathways get much larger as we go from arteries to arterioles. These are very, very small in diameter. You can barely see them with the naked eye. And they've got this really thick layer of smooth muscle, which is going to clamp down. So the function of arterioles, the arterioles function as resistance vessels. If we have high resistance in the arterioles, what's going to happen to the pressure? Well, the flow is going to have to remain consistent, right? Because we have a closed system. So we have 5 liters of blood that are going from the heart through the arteries to the arterioles. So the flow of blood has to remain consistent, but because the resistance changes, that's going to influence the pressure. So the pressure is going to get stepped down. And of course, there are many parallel pathways. Each arteriole is functioning somewhat independently. We'll talk about that in a minute. So what we see if we look across arterioles, we would see this maintenance of the systolic diastolic, which is an attribute of the arteries, the distensibility of the arteries, and the passive and active pushing of the arterial walls on the blood pressure, which is maintaining the diastolic pressure. We see the systolic diastolic, the pulse pressure difference, declining as we go through arterioles. Why is that? Partly because that smooth muscle, there's that myogenic effect. If you stretch the smooth muscle of the arterioles, it's going to clamp back, and it's going to go back to its original size. So with each heartbeat, you're going to stretch that arteriolar smooth muscle. It's going to clamp down, and it's got a really thick layer relative to its diameter. So it's going to really clamp on the pressure surge, and it's going to dissipate that pressure. So that what leaves the arterioles into the capillaries is just an ooze of blood that you can see is much lower pressure. Remember, the capillaries are very, very delicate structures, and they wouldn't be able to withstand a high pressure because they're just those endothelial cell layer, a single endothelial cell layer. So the arterioles, one of their major functions is to clamp down and eliminate the pulse pressure so that you do just get this ooze of blood going into the capillaries. Okay. How does this happen? How do the arterioles wind up accomplishing the step down of pressure? Well, we're going to find out that we have 3 different mechanisms that are employed, and they're all employed at the same time. The first is one that we've talked about, this myogenic regulation, or the auto-regulation of pressure by any layer of smooth muscle. And if smooth muscle is stretched, it contracts. This is exactly like we talked about in the arteries, but the arteries are these very large diameter, so the relative change in radius isn't going to influence the resistance all that much. Remember, arteries are very low resistance because they're huge diameter. Arterioles are much smaller diameter, so if we influence the radius, it's going to have a much more profound effect on the resistance. So this is something that has nothing to do with feedback by the nervous system or the endocrine system. It's simply a function of smooth muscle in itself. The second method that we have of regulating the diameter of arterioles has to do with the autonomic nervous system. And let's just review this. So the autonomic nervous system, remember, this is we have the sympathetic and the parasympathetic branch. We're going to focus just on sympathetic when we're talking about arterioles because we don't have any parasympathetic input to the arterioles. So the sympathetic branch, remember, this involves alpha-1, alpha-2, beta-1, beta-2 receptors. We're not going to talk about alpha-2 receptors in this scenario. Why is that? Because alpha-2 receptors are primarily in the digestive tract, in the organs of the digestive tract. They don't really influence the circulatory system at all. So we're going to talk about alpha-1, beta-1, and beta-2 receptors in this conversation. And all these receptors are activated by norepinephrine or epinephrine. Norepinephrine being the neurotransmitter primarily, epinephrine being the hormone that's released from the adrenal medulla. So let's talk about the alpha-1 receptors. Let's remember about them. So remember, the alpha-1 receptors preferentially bind norepinephrine. They can also be bound by epinephrine, by the hormone. but they preferentially bind epinephrine. They're excitatory. They activate a GS protein in the IP3 pathway to bring about their effect. These alpha-1 receptors are present in smooth muscle of arterioles across the entire body, with one big exception, except to the brain. We don't have those alpha-1 receptors on the arterioles that are carrying blood to the brain. Stimulation of these causes the arterioles to contract. We're going to activate, and we're going to show this in the next slide, how the IP3 pathway brings about constriction. If you constrict the arterioles, what's going to happen? The diameter is going to decrease a little bit, the resistance is going to go up a lot, and blood flow is going to decrease pretty profoundly. So this sounds a little counterproductive. We talked about this a couple weeks ago, that stimulation of the alpha-1 receptors during a sympathetic response, a fight or flight response, it seems that you want to increase the blood flow to the tissues of the body, when in fact the alpha-1 receptors are limiting the blood flow. So we'll get back to that in just a little bit. If we step up the resistance, what's going to happen to pressure? If resistance goes up, pressure is going to go up. So this is one mechanism by which the body is regulating blood pressure during a sympathetic response. We're going to find out it's not the only thing that's going on. There's something else very important going on, but this is maintaining the pressure. So while we're focusing on this constriction of smooth muscle, let's talk about the mechanism. And this is stepping back a little bit. This is going back to chapter 8 a little bit. But smooth muscle cells, we've talked about a little bit, are spindle-shaped, and they're called smooth muscle because they don't have that repeating sarcomere arrangement that we've talked about in striated muscle, in cardiac or in skeletal muscle. So they contain thick and thin filaments that are made up of actin and myosin, very similar, not exactly identical, but very similar to striated muscle. But the thick and thin filaments don't have that regular repeating pattern. And in fact, the thick and thin filaments are not arranged along the length of the muscle cell. They're arranged sort of in a crisscross pattern. So there's sort of this checker pattern of the arrangement of the thick and thin filaments. So when you look at a smooth muscle cell under a microscope, you don't see that A-band, I-band repeating business that we talked about previously, which is why they were originally called smooth muscle, because they didn't show that striation. Contraction of these thick and thin filaments is going to lead not to the shortening of the muscle, but a bunching up of the muscle. It's going to pull in all directions. And this big spindle is going to pull up into a little dimpled spindle. We have a similar mechanism of thin filaments that are made up of actin molecules and thick filaments that are made up of myosin molecules. But one big difference is that where we don't have troponin and tropomyosin that are inhibiting the thin filaments like we have in striated muscle. Remember, in skeletal muscle and in cardiac muscle, the thin filaments were coated with that tropomyosin, which is inhibiting the ability of the myosin heads to reach up and bind to the actin. In smooth muscle, there is no regulation on the thin filaments. The regulation is on the thick filaments, on the myosin heads. The myosin heads have to be phosphorylated in order to activate them to be able to bind to the thin filaments. So that's one major difference between thick and thin, between a smooth and striated muscle. So let's think about how this contraction is brought about. And to do that, we have to think way back to chapter 4. I know this has taken us back a lot of weeks, but remember the IP3 pathway of the second messenger systems that we talked about. So in the IP3 pathway, we have a surface receptor, a G-coupled receptor that is going to be activated by something. This is a very general figure, so we don't know exactly what's activating it yet. So this G-coupled receptor, when it gets stimulated, when it gets activated by whatever the appropriate stimulus is, it's going to activate a G protein. And in this case, it's a GS protein, a stimulatory protein. So that alpha subunit is going to come off. It's going to activate phospholipase C, which is going to split one of the phospholipids from the cell membrane into IP3 and diacylglycerol, and we're going to get this cascade event. Again, that's very regulatable, and it's also amplifying. We talked about second messenger systems a couple weeks ago. That was on the first exam, so I'm sure it's been purged out of your brain. But the reason that I bring this up is that this mechanism, this IP3 pathway, is the mechanism by which smooth muscle contracts. So let's take this now to the level of smooth muscle. So the surface receptors, there are a whole bunch of different surface receptors on smooth muscle. Remember, smooth muscle may be activated by stretch. So there may be stretch receptors that activate, that are G-coupled, and they're going to lead to the contraction mechanism that we're going to talk about next. These may be hormone receptors. they may be paraffin receptors. There are a whole bunch of different receptors that are present that are G-coupled in smooth muscle that are going to lead to cellular contraction. So the rest of the story for smooth muscle contraction involves this cascade, this IP3 cascade going on here. And if you follow down, I know that this is small on the screen and on your handout, but you'll see that calmodulin is one of the intermediates of the IP3 pathway. And this is going to lead to phosphorylation of some specific protein, which is going to bring about a cell response. In the specific example of smooth muscle, calmodulin is going to get activated by the calcium, which is the second messenger in the IP3 pathway. That's going to lead to activation of a protein called myosin light chain kinase. And myosin light chain kinase, when it gets activated by calmodulin, is going to phosphorylate the myosin heads, and that's going to allow the myosin heads then to interact with the actin.